Right. Well, hello everyone. Welcome to today's webinar, the Anthocyanin Enigma. My name is Elizabeth and I will be your moderator for today. I work on the marketing here at Anagenix and I'm excited to be hosting today's session. Before I introduce Doug, our speaker for today, I just hope that everybody is well out there and coping all right with the COVID-19 situation in your home country. Like many of you, we are also working from home. You probably would have heard that New Zealand is in lockdown. So it's not easy, I know, to keep the children quiet or to keep the dog quiet, but we are all doing the socially responsible thing. So just do not be surprised if you see a cat's tail popping up on our screen or if you hear one of our dogs barking. So right, so some of you may have met Doug in our last webinar on kiwi fruit pectin. For others who are seeing him for the first time, Doug is our product champion here at Anagenix. Doug helps to communicate the biomedical science behind Anagenix products. He has over 20 years of postgraduate research, publishing, and student supervisory experience in academia and industry. Doug holds a PhD in engineering and technology from Massey University and a Master of Science degree in Biological Sciences from the University of Auckland. So before I hand the mic over to Doug, just a few housekeeping items to cover about this presentation at webinar platform. So first, today's webinar will be available on demand after the live session and is accessible through the same link you're using now. We also have started a blog page on BerriQi. So if you go to berriqi.com, you would see our blog, and we will be posting this video on the blog page as well. So that will happen. We should probably have it up by next Monday, if not Tuesday. All right. You also may download today's presentation handout from the handout tab at the bottom of your screen. And we would also like to hear from you. So if you have questions for Doug, please feel free to send it through the Q&A tab in your menu bar. We will be answering questions at the end of today's session. So if we do not get to your question during today's webinar, we will definitely follow up with you afterwards, okay? And we also have a short survey at the end of the session, and we would like to hear from you, so please fill that out so that we can keep getting better at this. So last, we'd like to encourage you also to share today's webinar with your social networks. You can use the social media sharing icon on the top right of your menu to post this webinar directly on Twitter, Facebook, or LinkedIn. So without further ado, I'd like to kick things off by welcoming Doug. Doug, over to you.
Okay, thanks, Elizabeth. So, as the title says, we're going to talk about anthocyanins. Now, obviously, this webinar is to do with our respiratory health, which is one of our key health platforms here at Anagenix, and in particular, this webinar relates to BerriQi because it's got anthocyanins in it. As you can also see, we have products for digestive health and other products in the pipeline which cover other health platforms such as blood glucose management and things like that. So if you've got any interest in those areas, please do reach out to find out more. You can get in touch with us through the channels that brought you here or through my email at the end of this webinar. Right, for now, let's focus on BerriQi. So BerriQi assists with respiratory health. It helps our lungs, particularly in lung repair after inflammation. And one of the reasons it does this is because of the anthocyanins in it. So in this topic, we're going to cover anthocyanins. Firstly, BerriQi has them. Then we'll move along to what they are, what they do, and how and why they are good for us. So BerriQi contains anthocyanins and a bunch of other polyphenols and antioxidants which help keep airways clear. So we've done a lot of science to help show how BerriQi maintains healthy lungs, aids in reducing excessive mucus buildup, supports lung tissue repair and renewal, and assists with a healthy immune function, probably working through innate inflammatory pathways. BerriQi contains these polyphenolic bioactives, which in partnership with Plant and Food Research, who do a lot of research in partnership with us, we've looked at the composition of this. Now, anthocyanins are the major polyphenol found here. So and they come from boysenberry. So boysenberry supplies the anthocyanins, the hydrolyzable tannins, and half of the flavonols found in this. The other contributor to BerriQi apple supplies the phenolic acids, the chalcones, most of the flavin-thiols and procyanidins, and half of the flavonols. But for the rest of this talk, we're going to focus on that major component, the anthocyanins. This is what anthocyanins are. They're coloured compounds from plants, often edible plants. They come in a range of colours along the spectrum from red through blue, purple to black. And these are some of the edible plants that you're no doubt familiar with that are actually coloured by anthocyanins. Note that they're responsible for the colour in berries, that's true berries, and in the composite triplet fruit we also think of as berries, such as raspberries, blackberries, loganberries, and of course boysenberries, which are the berry component in BerriQi. Now, what do these anthocyanins do? Well, plants go to all the trouble of making these anthocyanins, not just to look nice, although that actually helps, but one of the reasons is because they're antioxidant. They protect plants from oxidative stress. So if you see on the circular images on the top right there, there's a nice green, healthy looking Arabidopsis plant, and on the right is one that's subject to an enormous amount of oxidative stress, and it's produced a lot of red and purple anthocyanins to actually protect from the stress. Another thing they do is attract pollinators. So bees and other insects are attracted to the colour of flowers, which is one of the reasons why flowers are actually coloured. It is in fact to look nice, but it's to look nice and attract insects. And of course it's to attract animals for seed dispersal, because they'll come along and recognise the colour of this fruit and recognise that it's ripe, and then they'll eat it and then excrete the seed somewhere else. And this is nicely illustrated by the civet here, who's eating these coffee berries of a very particular colour and ripeness, which will then excrete out later. And we all know this as coffee luat, and we'll pay enormous sums of money to get these coffee beans that have been dispersed by this animal because of their very exact degree of ripeness and consistency. So this is the chemical structure of an anthocyanin. Now, sorry for inflicting the chemistry on you, but it could be worse. It could be looking at this guy, which is a tannin also found in boysenberries. So here's our anthocyanin. It's a type of flavonoid, and I'll just go through and briefly describe some of its bits. This bit here is a sugar molecule, so it's that part is known as a sugar glycoside. This one happens to be glucose, and that hangs off the side of our anthocyanidin aglycone. That is the molecule lacking the glycoside. So this one in particular happens to be a cyanidin 3 glucose. Now, anthocyanins have got this characteristic 3-ring structure. There's an A ring, a B ring, and a C ring. It's the B ring hydroxylation pattern where a hydroxyl group is attached, by which the different types of anthocyanins are named. For example, this pattern is a cyanidin 3 glucoside, cyanidin because of the hydroxylation pattern of that B ring, and 3 glucoside because there is a glucose hanging off the 3 carbon of that central ring there. Now, this chemistry affects how they respond to pH, it affects their color, and a lot of their other attributes, including acidity, right? So acidity largely determines the color of anthocyanins, where they're more red under basic conditions, more blue in alkaline conditions. Okay, I'm actually going to illustrate this for you. I'll stop sharing my screen shortly. Let's see. If I do this, right, now I have 2 glasses of water, which I prepared previously. Into one of them, I'm going to make acidic with vinegar. Apologies you can't see this, but I'm limited by my working space here. And into the other, I'm going to make basic with baking powder. So I'll tip some of that in here. Right, so that's quite basic. I really don't want to drink that. And I also conveniently have here some BerriQi filled with anthocyanin. Now, BerriQi is naturally acidic at a pH of 3.4, so it'll come out naturally red. So if I take our effective dose of just over 5.5 mil, 7.4 grams, there we go. And I've been dripping this into my acidic solution. There we see that it goes quite red. I'll just put this over here, and I can illustrate that color by dipping a wick into it. See there, it has actually gone red. I'll just get rid of that. Now, next to this, I have my basic solution. I'll get a fresh spoon for this. Sorry, this one isn't transparent. I'll pour some more of our color into that. This is a nice red one, but as it goes into my alkali solution, you'll see that it's gone quite purple. It's quite a thick color. So what I'm going to do is also dip this into there, and you can see the difference. That is quite purple. That's the difference pH makes to this with the structure of anthocyanins. Now, to share my screen again. I assume you are seeing the appropriate screen. Good. Here again, so I've illustrated the color range, and also you can see the different names of the different anthocyanins, again attributed by the B ring hydroxylation. Now, what happens when we eat these anthocyanins? Well, so if we take our berries, here we have our poison fruit, abundant in red anthocyanins, and then when we eat them, they will be broken down into smaller molecules. So some of the molecules shown there are, for example, the PCA and PGA, and ultimately traveling through things like vanillic acid, the VA, and 4-hydroxybenzaldehyde. Now, again, sorry for the chemistry, but it could be worse. Could be one of these things. Again, why am I showing you this? Well, because if we highlight this region, we see that there's actually a repeating motif here. This is how biology works, in that there's this sort of very modular structure that happens with these plant polyphenols. So going back to our polyphenol breakdown products, we have our PCA, our PGA, our vanillic acid, our 4-hydroxybenzaldehyde. These will circulate in the body, exerting their bioactive effects, and ultimately they'll be converted to these molecules, which will then be disposed of. So we've got hippuric acid on the left, which is how most plant polyphenols will be excreted in the urine. It's named because it's actually named after horse urine, discovered no doubt when they were checking horses for doping. And on the right, we've got another end product, ferulic acid. Now, when we eat these anthocyanin-rich fruit, if we actually try and measure the availability of anthocyanins, they appear to not be very bioavailable, and they're not found in high concentrations in blood after eating. So previously, this has been thought that maybe they haven't actually been absorbed and they've just been excreted. But what actually happens is because their breakdown products are available. Now, this has actually been determined by a nifty little bit of science using some heavy isotope 13C tracer studies. This capitalizes on the fact that heavy carbon isotopes like 13C are 1 mass unit per carbon heavier than a normal 12C isotope. So on the right there, we see 12C with its 6 protons and 6 neutrons, making an atomic mass of 12. And then we've got our heavy 13C with its extra neutron. Now, if you synthesize or label a chemical compound with these, then chemically they are indistinct from their original compound, their unlabeled compound, and they'll behave the same way, so that you can actually you can detect them using their mass, but you can separate them using their chemistry. So what happens is participants were fed heavy labeled anthocyanin, they're cyanide 3-glucoside, and then their blood and urine was sampled over time, and then the anthocyanin and their breakdown products were separated by their chemistry, identified by their mass, and shown which parent compound they came from. What we find is that over time we see the appearance and disappearance of different products, And what happens is there's a peak time in which you see the time at which you see the most of this compound circulating through the blood. So what we see here in this figure is that over time, see at the top, a variety of things appear in different orders. So our cyanidin 3-glucoside, our parent anthocyanin, it maxes out at 1.8 hours and then declines thereafter, but it reaches a concentration of 141 nanomolar. Moving to our right, one of our end products, ferulic acid, it maxes out at 8.2 hours in the body, but we'll see directly below it has a much higher concentration, 827 nanomolar. Then at 12.5 hours, we're getting a peak of vanillic acid as a breakdown product of the anthocyanin, and there's a lot more of this. There's almost 2,000 nanomolar in the blood. After 15 hours, we start seeing hippuric acid, and after 30 hours, we're seeing vanillic acid sulfate. So these are vanillic acids, which our body is now sulfated and targeted for disposal. Ultimately, they too will be turned into hippuric acid and ferulic acids. Now, there's the times when they actually reach peak concentration, but it's all about how much time do they spend in the body overall. As you can see so our cyanidin, it has a really short half-life. So by 0.4 hours, half of it's actually already gone. So it peaks a little bit later, but it does have a fast breakdown rate, which is consistent with the amount of byproducts we start seeing. After 6.5 hours, we start seeing that that's our half-life of vanillic acid. So it lasts a lot longer than the cyanidin, but it's quite short. Obviously, if we go right to the end to our hippuric acid, that's got a really long half-life because essentially it's an inactive breakdown product that's just hanging around for disposal. And if you look under the bar there, you'll see that the concentrations tell a real story. The concentration of the anthocyanin is really quite low. This is the total amount, the area under the curve, the total amount that the body has seen over time. So things like our vanillic acids and our vanillic acid sulfates and our ferulic acids, the body sees a lot more of those than it actually does anthocyanin. So we have to consider that potentially any bioactivity we see from the consumption of anthocyanins actually comes from these breakdown products. So why is there such a difference in the peak times and half-lives of these circulating bioactives? Well, first, they have to be absorbed in the gut. Some aren't. They'll just completely pass through the gut and be excreted in feces. And then those that are absorbed in the gut, some are absorbed in our small intestine after interacting with our digestive enzymes. Some of them survive beyond that and then interact with the gut bacteria which reside in our colon, and there they'll be broken down into these smaller parts which are then absorbed and make it into the body. Right, if we see on the figure on the right, so from the gut, then they will circulate through the portal vein, that blue bit, to the liver, and from the liver, they'll then return through the venous system to the heart, and then they'll go through the lungs, and then ultimately through to the right, left side of the heart, where it'll then be distributed to the rest of the body. So it goes through from the gut, it goes to the liver. We'll get that first pass metabolism, where some more breakdown occurs. Then it circulates through the lungs, and then through the rest of the body, and eventually going through the rest of the body means it passes through the liver again as a second pass metabolism. This is where additional breakdown happens, where things are targeted for disposal, and ultimately it'll all be filtered out by the kidneys into the urinary system. Now, there is a feature here that's actually quite important. We'll come back to this later. The fact that after it goes from the gut, passes through the liver, via the heart to the lungs before the rest of the body. We're going to come back to this later. Now, what do anthocyanins actually do? We've talked about them being biologically active. Again, the scientific literature is not always clear whether it's the anthocyanin itself, its byproducts, those intermediate breakdowns products, the PCA and PGA, or the endpoint products such as the ferulic acid or hippuric acid. But irrespective of that, anthocyanins and their intermediates are chemically antioxidant, so they will directly soak up free radicals, reactive oxygen species, reactive nitrogen species, and so on, and they will just act as antioxidants. They'll also turn on genes for antioxidant response elements. These are our body's defenses against free radicals, such as superoxide against reactive oxygen species, superoxide dismutase against superoxide, which is a reactive oxygen species, or catalase, which is active against peroxide. Now, this sugar hanging off the side of the anthocyanin may interact with digestive enzymes because our digestive enzymes recognize the sugar. This interaction may in fact slow down our digestion of other sugar-containing compounds like sucrose or starch, and that may actually modulate our rate of digestion of other foods. Anthocyanins and some of their byproducts, which have been absorbed by cells, have been shown to interact with this protein, well, composite protein, which is called nuclear factor kappa B. Now, NF-kappa B is a transcription regulator. It's shown here on the right, interacting with DNA. So transcription regulators alter gene expression. Now, remember, gene expression is what happens when you want to turn a gene into an effect. So our gene expression results in production of messenger RNA. Messenger RNA is then transcribed into proteins. Proteins are what do stuff in the cell. NF-kappa B increases the expression of genes involved in inflammation, these pro-inflammatory genes. So anthocyanins and some of these byproducts, they bind to NF-kappa B and they weaken or stop it interacting with DNA, thereby decreasing inflammation. And so anthocyanins and their breakdown products are potentially anti-inflammatory through that mechanism. Now, anthocyanin and some of its byproducts will also interact with another type of protein called a Janus kinase, which is it's a kinase, as I said, so it phosphorylates other proteins, and also with signal transducer and activator of transcription 6, known as STAT6. Now, our Janus kinase receives us signals from cell receptors. There's a conformational shift which activates the Janus kinase, and that will then these two will phosphorylate each other and activate STAT6. STAT6 will activate the expression of this gene GATA3. This GATA3 protein then in turn promotes expression of the pro-inflammatory cytokines interleukin 4, interleukin 13, and interleukin 5 listed here. Interleukins 4, 13, and 5 are pro-inflammatory cytokines. So they have been implicated in things like atopic disease, allergy, asthma, dermatitis, these sort of things. Excuse me. Now, because these anthocyanins and some of their byproducts inhibit this JAK-STAT signaling pathway, or signaling cascade as it's known, which is intended to result in production of these inflammatory cytokines, which are messengers to promote inflammation. Therefore, anthocyanins are decreasing allergic inflammation. Now, returning to that point before, anthocyanin bioactivity and where they go. Remember, the absorbed bioactive from the gut will travel via the liver and pass through the lungs, the pulmonary circulation, and back to the heart before circulating through the rest of the body. So if you watch the image here, bioactives go through the gut, then they'll travel to the liver, then through the heart to the lungs, where the blood's also getting oxygenated, and now that blood is traveling through to the rest of the body. What this means is it makes the lungs ideal targets to study the bioactivity of anthocyanins and first mass metabolism, because lungs receive first cut at them. It also means that there's a lot of potential of looking at anthocyanins and other bioactives as lung health products. So this is what researchers at Plant and Food Research have done. They've shown that BerriQi, which is our drink rich in anthocyanins, it improves small animal lung function after an acute exposure to urban dust particles. So what happens here is the animals are primed, so their immune system is active, and then they're exposed to these urban dust particles intranasally so that they'll enter into their lungs and stimulate this acute inflammation. And then they'll consume a measured amount of BerriQi rich in anthocyanins, and the amount of anthocyanins is particularly standardized dose. And then at the end of the experiment, lung tissues will be collected and analyzed after the animals have been assessed for their breathing abilities. And we can go through and show you those results. So what this first graph shows is how this BerriQi rich in anthocyanins relieves lung inflammation. So the vertical axis shows the total number of immune cells recovered from a bronchial alveolar lavage fluid. So this is actually just a liquid rinse of the lungs, and immune cells are recovered from that and measured especially. And what we find is on the pink bar on the left, our naive mice, which have received no allergen, have the lowest number of infiltrating immune cells. Our ones which have received this urban dust stimulus, their lungs are now inflamed and they have a much greater number of immune cells. Those that have received this inflammation and have also consumed this BerriQi rich in anthocyanins show a much reduced number of immune cells in their lungs. So there's that anti-inflammatory effect we talked about before. What we're also finding is that it's improving lung function. Now it's relieving constriction. If you think of constriction as the tightening of a fist or something or other, our naive mice have the lowest constriction amount, so their lungs are able to relax. Our dust stimulated allergic mice have the highest constriction of the lungs, and those that have consumed our anthocyanin-rich BerriQi now have a much reduced constriction. It's almost back to our sort of normal levels. We also find that our naive mice have the greatest ability for their lungs still maintain that ability to stretch because they're not inflamed. Those with dust-induced inflammation of the lungs have a lower lung stretch, and that is restored when they consume BerriQi rich in anthocyanins. And here we've got measures of lung inspiratory capacity. That's the ability to breathe in after a normal breath out. Our naive mice, of course, have the highest lung inspiratory capacity. Our dust-induced inflamed mice have the least, and those that have consumed BerriQi rich in anthocyanins have that somewhat restored. Also, BerriQi rich in anthocyanins has been confirmed to work in a acute inflammation model of allergic asthma. In a chronic inflammation model, well, this is where there's ongoing inflammatory stimuli leading to this ongoing chronic inflammation. And what we know there is, using similar experiments, that this BerriQi with anthocyanins will reduce immune cell infiltration into the lungs, like the example you saw there. It'll also reduce that mucus hyperproduction, and it will activate a repair mode in macrophages, which are a particular type of immune cell which get involved in things like wound healing and soaking up damaged tissue or invaders, and ultimately reduce lung fibrosis in this chronic inflammation model. And the science behind that is going through the publication process as we speak, so will become available. So in summary, our anthocyanins, they're coloured polyphenols from plants. They've got different forms due to their different chemistry, and they have different bioavailable byproducts as they travel through our digestive tract and are broken down. These anthocyanins and these byproducts have different concentrations and half-lives in the blood, so their concentration peaks at different times, and the length of time they stay in the blood, or the rate of their decline, is quite different. Anthocyanins and their breakdown products are chemically antioxidant, and they'll regulate antioxidant gene expression. They also control pro-inflammatory gene expression, so they can inhibit that through multiple mechanisms. And they've been experimentally shown in a preclinical animal model that they'll modulate airway inflammation. They can activate a post-inflammatory repair response. So there we have it, BerriQi with anthocyanins containing your daily dose for mitigating airway inflammation. And that's the end of our webinar. If there are any questions, hopefully you will have shot them through where Elizabeth can take over and read them out.
Well, thank you, Doug. That was a good presentation, and
Very
interesting about byproducts of anthocyanin, as well as the lung being the first organ the anthocyanins after the first and second bypass from the liver.
I'm sorry, Elizabeth, I can't hear you. I think we're having some streaming problems with our internet. I'm not sure if this is me or everyone else is receiving this.
We have a couple of questions here. I'm just going to give this question here. So the first question I think is quite a good one. How is the anthocyanin very different from the anthocyanin?
I'm sorry, Elizabeth, I can't hear you. Can you say that again, please? Can you hear me?
Yeah. Are you able to hear me?
Yes, the line seems to have cleared now.
Yes, I can hear you.
Yeah.
Okay. Okay, so what I've done, I'm closing off my video just so that hopefully this will work better, okay? So here's the first question. How is the anthocyanin, okay, how's the anthocyanin in BerriQi different from anthocyanin in purple or red fruits?
How is it different? Lots of fruits do contain anthocyanins at quite high levels. I think the difference is what we've done is we've actually checked their function, and we've shown that the particular mix of anthocyanins in the particular proportions we've got, and in combination with the apple juice we're using, really works well against lung post-inflammatory repair.
Okay, right, that's good. This is another question, and it's coming from actually from Thailand, and they probably and they are familiar probably with apple and BerriQi. So that question is, what is the benefit of having apple in BerriQi, and what's the synergy between apple?
Yeah, well, Boysenberries got a distinctive, quite tangy taste. So apple was one of many fruit that was explored to combine with that taste. And it was actually found that the apple polyphenols, particularly things like the procyanidins, they actually combine with the boysenberry anthocyanins and other compounds to actually increase the effect we see, particularly against eosinophils, which are a class of immune cells found in allergic asthma. And some of these apple compounds have separately been shown to actually target some of these pro-inflammatory pathways or immune cell recruitment factors. So apple and boysenberry together appear to function better than boysenberry alone or apple alone.
Okay, thanks, Doug. This is a question about the breakdown of compounds of anthocyanin, the byproducts. So here's a question: does it mean that the end breakdown product in anthocyanin in BerriQi is hippuric acid, and it takes around 90 hours?
Ultimately, that will be one of two endpoint products that have been measured by chemistry. So what will happen is the anthocyanins will initially be broken down into things like the proto-catechuric acid and fluoroglycerol excuse me if I've mangled the names there. And those in turn will be break down into other products, such as the vanillic acids and the vanillic acid sulfates and a bunch of other compounds. And ultimately, those in turn will break down into hippuric or ferulic acids. And there's a couple of nice papers in the literature where people have actually tracked these with those isotopes, and they can show which molecule turned into which.
Okay. So here's another one. Is anthocyanin the only active compound to make BerriQi effective?
It's unlikely that they're the only one, but given that they are the major bioactive and there's so much science behind them, it's likely that they are responsible for most of the effect. Also, we have data on a heat inactivated BerriQi where when you look at the chemistry, the major compounds which are missing are the anthocyanins, and that heat inactivated BerriQi failed to show any of the lung benefits of the unheated version. So we think it's likely that anthocyanins are responsible for most of the effects, with of course a contribution with some of the other polyphenols from the apple.
Right, that's good, Doug. I'm just reading one other question here. I will just read that. If anthocyanin gets broken down prior to reaching lungs, if it went direct to the lung, would you have a greater effect, or do you need it to be broken down before it hits the lungs?
We just don't know. What we do know is that it'll, after that first pass metabolism through the liver, and obviously being absorbed through the gut wall in that process, it will go to the lungs before it's distributed to the rest of the body. So your lungs are going to see it before your brain does, before your arms and legs do. And given that you do measure it, you still do measure anthocyanins after 1 or 2 hours in the blood. The lungs do see anthocyanins.
Okay, that's good. So we have a flurry of questions
I'm not
this
sure I
morning.
answered the question, but yeah.
Well, we can pick this up. So if any of you want further clarification, please drop us an email. Okay. What percentage of the anthocyanin or the breakdown products end up being absorbed?
I'm not sure that we can know that. That's sort of a mass balance experiment, which is quite hard to do. I guess one could conduct their own experiment by looking for coloured compounds in what they excrete after consuming it. But yeah, we just don't know. It's likely most of it, based on previous papers.
Right. Okay. This one is an interesting question. It's really very close to everybody's heart at the moment, you know. So how can anthocyanin help with the current COVID-19 situation?
Okay. Well, we don't have data to show that anthocyanins or the other polyphenols in BerriQi can actually act directly against viruses. What we do know is that the anthocyanin-rich BerriQi does appear to activate post-inflammatory repair mechanisms. So certainly if one is clear of the virus but suffering from chronic inflammation or post-inflammatory damage, we'd recommend you take it, but we can't actually advise anything to do with viruses.
Okay. Right. I think that's all the questions. Let me just check on the board that we've covered them.
There's some tricky ones there.
Yeah, but good questions from everyone. Good response. Okay, so I think we've covered most of the questions. Well, thank you everyone for your participation this morning. And so we have a paper that we've done on a deep dive on anthocyanins and also on lung health and how BerriQi help with that. So if you're interested, just write to Doug or myself and we'll send that off to you. And again, this webinar will be available on berriqi.com blog page. So keep a lookout for it. We will get it up by Monday. Yeah, and in the meantime, please keep safe, and we are sending lots of positive energy to you. Okay, so thank you and have a wonderful weekend, everyone. Bye-bye.